Hi viewers, welcome back again to one more other video session from this channel. So in this video session we will be studying regarding endomembrane system and as well as subcellular organelles in detail. So here what is endomembrane system and what are the various subcellular organelles which are most commonly present within the cytoplasm of the eukaryotic cell. So here, as I have already mentioned you, within the cytoplasm of the eukaryotic cell, here we can observe many of the subcellular organelles. So here what are the various subcellular organelles which are most commonly present within the cytoplasm of the eukaryotic cell? Here the various subcellular organelles include endoplasmic reticulum, Golgi complex and as well as lysosome. And in addition to these specific subcellular organelles, we can observe some more other specific subcellular organelles like ribosomes, mitochondria and as well as peroxisomes. And especially in case of plant cells, we can observe some more other additional subcellular organelles like plastids and as well as vacuoles. Is it clear? So here all these are the various subcellular organelles which are present within the cytoplasm of the eukaryotic cell. So let me write it here. Subcellular organelles. So here the various subcellular organelles include endoplasmic reticulum, Golgi complex, lysosome, and the other organelles include ribosomes, mitochondria, peroxisomes, and especially in case of plant cells we can observe plastids and as well as vacuoles. So here all these are the various subcellular organelles which are most commonly present within the cytoplasm of the eukaryotic cell. So here out of these many subcellular organelles, the specific subcellular organelles which are responsible for the formation of endomembrane system, it includes endoplasmic reticulum, Golgi complex, lysosomes and as well as vacuoles. So here all these specific subcellular organelles, they are responsible for the formation of endomembrane system because all these specific subcellular organelles, they are interconnected with each other. Is it clear? They remain in an aggregate form. So the reason why they lead to the formation of endomembrane system. Endomembrane system, right? Endoplasmic reticulum, Golgi complex, lysosome and as well as vacuoles. So here all these are the various subcellular organelles which come under the endomembrane system. Then what are the various subcellular organelles which do not come under the endomembrane system? Here the various subcellular organelles which do not come under the endomembrane system, they include chloroplast, peroxisomes and as well as mitochondria. Chloroplast, peroxisomes and as well as mitochondria are not involved in endomembrane system. Endomembrane system. So at first we will be studying regarding endoplasmic reticulum. So here what is endoplasmic reticulum? Endoplasmic reticulum, it is the largest subcellular organelle which is most commonly present in case of eukaryotic cell. Is it clear? So let me arrange the things first. And if we consider the endoplasmic reticulum, so here within the cytoplasm of the eukaryotic cell, here we can observe many number of network tubular like structures which are scattered within the cytoplasm of the eukaryotic cell. So here these specific tubular-like structures which are scattered within the cytoplasm of the eukaryotic cell, here it is termed as what? Endoplasmic reticulum. Endoplasmic reticulum, it is the largest subcellular organelle which is present within the eukaryotic cell. Is it clear? Endoplasmic reticulum is a network of tubular structures which are scattered within the cytoplasm. So here endoplasmic reticulum, it is the largest subcellular organelle which is most commonly present within the eukaryotic cell. And here we can classify the endoplasmic reticulum into two different types simply basing on the structural organization. That means if on the peripheral region of the endoplasmic reticulum, if membrane bound ribosomes are present, presence of membrane bound of ribosomes on the peripheral region of the endoplasmic reticulum, it induces a rough appearance or a zigzag appearance, and here it is termed as what? Rough endoplasmic reticulum. Then in the same way, if on the peripheral region of the endoplasmic reticulum, if there are no membrane bound ribosomes, it induces a smooth appearance or a glittering appearance or a shining appearance, and here it is termed as what? Smooth endoplasmic reticulum. So let me write it here. Endoplasmic reticulum, it can be classified into two different types, and these include smooth endoplasmic reticulum and the other one is rough endoplasmic reticulum. So here what is the function of smooth endoplasmic reticulum and as well as rough endoplasmic reticulum? At first, if we consider the function of rough endoplasmic reticulum, so most commonly the membrane bound ribosomes which are present on the peripheral region of the endoplasmic reticulum, they are responsible for the biosynthesis of protein. Is it clear? Rough endoplasmic reticulum, it is involved in the biosynthesis of proteins. But whereas if we consider the function of smooth endoplasmic reticulum, most commonly smooth endoplasmic reticulum, it is responsible for regulating the lipid biosynthesis. That means the specific lipid molecules which are responsible for the synthesis of cell membrane or plasma membrane, most commonly they are synthesized within the smooth endoplasmic reticulum. Is it clear? And in addition to this, even the particular smooth endoplasmic reticulum, it is responsible for regulating the drug detoxification. In order to detoxify the toxic substances from the body, the smooth endoplasmic reticulum, which is a part of the liver cells, it plays an important role. Is it clear? So here this is regarding drug detoxification, and in addition to this, even the particular smooth endoplasmic reticulum, it is responsible for regulating the biosynthesis of steroid hormones. So here all the steroid hormones like testosterone, estrogen and progesterone and all the other steroid hormones, they are successfully synthesized within the smooth endoplasmic reticulum. And one of the modified form of smooth endoplasmic reticulum, here it is nothing but what? Sarcoplasmic reticulum, which is most commonly available or which is most commonly observed in case of skeletal muscles. It is responsible for the biosynthesis of calcium. Is it clear? So let me write it here. Smooth endoplasmic reticulum, it is responsible for regulating lipid biosynthesis, drug detoxification, calcium regulation. So here the calcium regulation, it is successfully organized by the means of the modified form of smooth endoplasmic reticulum, which is nothing but sarcoplasmic reticulum. And in addition to this, even the smooth endoplasmic reticulum, it is responsible for the biosynthesis of steroid hormones. Is it clear? So here this is regarding endoplasmic reticulum. Then after we will be studying regarding Golgi complex. Is it clear? So here what is Golgi complex? So let me arrange the things first. If we consider the Golgi complex or it is otherwise known as Golgi body or Golgi apparatus. So here all these are the various synonyms. So here the particular Golgi body or the Golgi apparatus, it was observed by a well-known Italian cell biologist, Camillo Golgi. So here Camillo Golgi, for the very first time, he observed certain reticular-like structures which are present very near to the nucleus by taking the advantage of certain staining procedures. So here within the particular Golgi complex or the Golgi apparatus, many number of thin flattened tubular cyclic structures are present, and here they are nothing but what? Golgi cisternae. Is it clear? Within the particular Golgi complex or the Golgi apparatus, here we can observe many number of thin flattened tubular cyclic structures, and here they are nothing but what? Golgi cisternae. And here the particular Golgi cisternae, they are present very adjacent to each other. Once when many number of Golgi cisternae, once when they undergo aggregation, they lead to the formation of what? Golgi stack. Is it clear? So here all these are nothing but what? Golgi cisternae. Once when all the Golgi cisternae, once when they undergo aggregation, they lead to the formation of Golgi stack. And here the number of Golgi cisternae, it remains vary in case of the various living cells. In case of all the living cells, the number of Golgi cisternae, it won't remain similar, it remains vary. So let me write it here. Golgi complex first observed by Camillo Golgi. Camillo Golgi. And here within the particular Golgi apparatus, thin flattened tubular cyclic structures are present. Cyclic structures are present. And here they are nothing but what? Golgi cisternae. And here the particular Golgi cisternae, it remains vary in case of the various living cells. In case of all the living cells, the number of Golgi cisternae, it won't remain similar, it remains vary. And here basically the particular Golgi apparatus, it has two different orientations, and these include cis Golgi and the other one is trans Golgi. Cis Golgi, it is responsible for inducing the entry, but whereas trans Golgi, it is responsible for inducing the exit. Is it clear? Cis Golgi, it is convex in shape, but whereas trans Golgi, it is concave in shape. So most commonly the particular Golgi apparatus, it is responsible for regulating the protein modification and as well as lipid modification. That means the specific proteins which are successfully synthesized by taking the advantage of membrane bound ribosomes, these specific proteins, they are successfully translocated within the particular Golgi apparatus for undergoing specific modifications. Is it clear? So let me show it here. So here let us assume it has the secretory vesicle. Within the secretory vesicle, receptors are present. Is it clear? To the concerned receptor, the specific proteins which are successfully synthesized by taking the advantage of membrane bound ribosomes, they successfully recruit here. So once when the receptor ligand complex, when it has successfully formed, now the secretory vesicle, so here it is nothing but what? The secretory vesicle. The specific secretory vesicle, it undergoes translocation into the particular Golgi apparatus by taking the advantage of cis Golgi. So here, once when the particular secretory vesicle, when it undergoes successful translocation into the Golgi apparatus by taking the advantage of cis Golgi, now the particular protein, it undergoes modification within the Golgi apparatus. And here the specific modification, here it is nothing but what? Glycosylation. Is it clear? Glycosylation. So here glycosylation, it is the protein modification which is observed in case of Golgi apparatus. In case of Golgi apparatus. So here, not only the specific protein, even the specific lipid molecules, they also undergo modification within the particular Golgi apparatus. In general, after the successful synthesis of the lipid molecule within the endoplasmic reticulum, now the particular lipid molecule, it undergoes successful translocation into the particular Golgi apparatus, and here the specific lipid molecule, it undergoes modification within the particular Golgi apparatus, and here it leads to the formation of what? Glycolipids. Is it clear? Both the proteins and as well as lipids, they are successfully modified within the Golgi apparatus, and in addition to this, even the particular Golgi apparatus, it acts as a major site for the biosynthesis of certain polysaccharides like cellulose. So let me write it here. In case of plant cells, Golgi apparatus is responsible for the biosynthesis of cellulose. So here the particular cellulose, it plays an important role in the formation of cell wall in case of plant cells, and in addition to this, even in case of animal cells, Golgi apparatus, they act as a major site for the biosynthesis of glycosaminoglycan. So here the particular glycosaminoglycan, it plays an important role in the formation of extracellular matrix. So here all these are the various important aspects of the particular Golgi apparatus. Then after we will be studying regarding lysosomes. Is it clear? So let me arrange the things first. And if we consider the particular lysosomes, so here the particular lysosomes, they are termed as suicidal bags of the biological cells. So here why the particular lysosomes, they are termed as suicidal bags of the biological cells? Because the particular lysosomes, they are responsible for the biodegradation of the various biological cells and as well as the particular biomolecules. So here in order to degrade the biological cells and as well as the biomolecules, within the interior side of the lysosome, many number of hydrolytic enzymes, they are present. Is it clear? Within the interior side of the lysosome, here we can observe nearly 40 hydrolytic enzymes. So here these 40 hydrolytic enzymes, they include lipase. So let me write it here. Lysosomes are considered as suicidal bags of biological cells. So here within the interior side of lysosome, nearly 40 hydrolytic enzymes are present, and these include lipase, some of the hydrolytic enzymes like lipase, protease, RNase and DNase. So here all these are the hydrolytic enzymes which are responsible for the biodegradation of the various biological cells and as well as the biomolecules. And here all these hydrolytic enzymes, they remain in active conformation only in the presence of internal acidic pH. Is it clear? So here all the various hydrolytic enzymes, they remain in active conformation only in the presence of internal acidic pH. So here in order to maintain the constant internal acidic pH, here the particular lysosome, it takes the advantage of a specific enzyme which is nothing but what? Vacuolar H plus ATPase. So here it is nothing but what? Vacuolar H plus ATPase. So here the particular vacuolar H plus ATPase, it is responsible for pumping the H plus ions or the protons against the concentration gradient. Is it clear? Here the concentration of protons, it remains higher within the interior side of the lysosome but not in case of the cytoplasm. So here once when the particular H plus ions or the protons, once when they are traveling against the concentration gradient or from the lower concentration to the higher concentration, they need the energy or they take the advantage of energy. And here the particular energy, it is provided by the means of ATP. Is it clear? So here the vacuolar H plus ATPase, it is responsible for pumping the H plus ions or the protons against the concentration gradient. So here within the presence of the internal acidic pH, all the various hydrolytic enzymes, they are capable to degrade the biological cells and as well as the biomolecules. Is it clear? So here this is regarding lysosomes. Then after we will be studying regarding vacuoles. So here what are vacuoles? And if we consider the particular vacuoles, So here vacuoles are the membrane bound space subcellular organelles which are most commonly present within the cytoplasm of certain living organisms like plants, fungi, and even in case of protists. Is it clear? So let me write it here. These are the membrane bound subcellular organelles. Subcellular organelles which are present in case of plants, fungi, and as well as protists. So here most commonly the particular vacuoles, they are enclosed by the means of a single most outer covering layer which is nothing but what, tonoplast. And here the particular vacuole, it is enriched with certain chemical constituents like water and as well as sap. And even the particular vacuole, it is also enriched with certain non-essential components which are a part of the particular plant cell. Vacuoles, they contain water, sap, and even it also contains certain non-essential components. So here the concentration of all these chemical constituents, they remain higher in case of the vacuole in comparison with the cytoplasm. So here in order to pump all these specific chemical constituents against their concentration gradient, here energy is required. Is it clear? All these chemical constituents, their concentration, it remains higher in the interior side of the vacuole in comparison with the cytoplasm. So here in order to pump all these chemical constituents against the concentration gradient, here energy is required. And most commonly, if we consider the particular plant cell as 100%, out of 100% of the particular plant cell, around 90% of the particular plant cell, it is occupied by the means of the particular vacuole. Is it clear? And we can classify the particular vacuoles into two different types, and these include contractile vacuole and the other one is food vacuole. Contractile vacuole and the other one is food vacuole. So here most commonly the particular contractile vacuoles, they are observed in case of certain unicellular organisms like amoeba, and here they are responsible for inducing the osmoregulation. But whereas food vacuoles, they are present in case of certain living organisms like protists, they are responsible for engulfing the food particles, or they are responsible for the storage of food particles. So let me write it here. Contractile vacuole, they regulate, they induce osmoregulation. But whereas food vacuoles, they are responsible for storing the food particles. So then after we will be studying regarding mitochondria. So here what is mitochondria? So let me arrange the things first. Mitochondria. So here the term mitochondria, it is derived from two Greek words, and these include mitos and the other one is chondrion. So here the meaning of mitos is thread and the meaning of chondrion is granules. So let me write it here. Mitos means thread and the meaning of chondrion is granules. So here the term mitochondria, for the very first time, it was coined by C. Benda. C. Benda, for the very first time, he coined the term mitochondria. So here the particular mitochondria, it can be visualized only once when the particular mitochondria, when it is stained by taking the advantage of certain staining procedures. So here after the successful staining of the particular mitochondria by taking the advantage of certain staining procedures, now the particular stained mitochondria, it can be visualized under microscopic observations. Is it clear? So here the number of the mitochondria, it remains vary according to the physiological activity of the particular biological cell. So let me write it here. Number of mitochondria remains vary according to the physiological activity. So here not only the number, even the size and the shape of the mitochondria, it also remains vary. Nearly in case of all the living cells, the size and the shape of the mitochondria, it won't remain similar, it remains vary. And here if we consider the diameter, at first if we consider the shape of the mitochondria, it can be either sausage shape or it can be cylindrical shape. And if we consider the diameter of the particular mitochondria, it ranges around 0.2 micrometers to 1 micrometer. 1 micrometer. And if we consider the length of the particular mitochondria, it ranges around 1 micrometer to 4 micrometers. And basically, if we consider the particular mitochondria, basically it contains two specific membranes. What are those? Outer membrane and the other one is inner membrane. Is it clear? So here in between the outer membrane and as well as inner membrane, here there is an availability of a specific space which is nothing but what? Intermembrane space. Intermembrane space, it is the specific region which is present in between the outer membrane and as well as inner membrane. So here both the outer membrane and as well as inner membrane, they are very specific in accordance to the particular enzymes. So here both the outer membrane and as well as the inner membrane of the mitochondria, they are having their own enzymes. Is it clear? So here the outer membrane of the mitochondria, it is nearly permeable nearly to all the solutes, but whereas the inner membrane of the mitochondria, it is very selectively permeable. It won't allow all kind of solutes to come across it. Is it clear? So let me arrange the things first. Outer membrane of mitochondria. Outer membrane allows all the solutes, but whereas inner membrane, it is selectively permeable. Selectively permeable, right? And in association with the inner membrane of the mitochondria, here we can observe a specific homogeneous substance which is nothing but what? Mitochondrial matrix. In association with the mitochondrial matrix, here we can observe certain inner folding like structures which are nothing but what? Cristae. So here these are nothing but what? Cristae. So here the particular cristae, they are responsible for increasing the surface area of the particular mitochondria. Is it clear? So here the particular mitochondria, they are the sites of aerobic respiration. That means within the particular mitochondria, the energy currency of the particular biological cell, which is nothing but ATP, it successfully synthesizes within the particular mitochondria. So the reason why mitochondria, they are the powerhouses of the particular biological cell. Is it clear? Even the particular mitochondria, they are the sites for aerobic respiration. So let me write it here. Mitochondria are the sites for aerobic respiration. And even mitochondria, they are termed as powerhouses of the particular biological cell. Powerhouse of biological cell. And here the particular mitochondria, they are termed as semi-autonomous organelles. So here why the particular mitochondria, here they are termed as semi-autonomous organelles? Because the particular mitochondria, they possess their own double-stranded circular DNA. Is it clear? Mitochondria themselves, they possess their own genetic material. and in association with the particular genetic material, even the particular mitochondria, they possess their own ribosomes. Is it clear? Mitochondria, they are termed as semi-autonomous organelles. Because the particular mitochondria, they has their own double-stranded circular DNA, and in addition, even the particular mitochondria, they has their own ribosomes. So here, in order to synthesize the particular protein, the particular mitochondria, they need not to take the advantage of the particular nucleus. Without the supervision of the nucleus, the particular mitochondria, they are capable to synthesize their own proteins, because the particular mitochondria themselves, they possess their own genetic material, and even themselves they possess their own ribosomes. And here the specific ribosomes which are present within the particular mitochondria, they are nothing but what? 70S ribosomes. They are not 80S ribosomes, they are 70S ribosomes. And if we consider the proliferative nature, likewise the particular bacteria, even the particular mitochondria, they are capable to induce the proliferation by taking the advantage of a specific mechanism which is nothing but what? Binary fission. So let me write it here. Mitochondria are capable to manipulate, or they are capable to increase their number by the means of binary fission. So likewise the bacteria, even the particular mitochondria, they undergo binary fission. Is it clear? So here this is regarding mitochondria, and in the next upcoming video session we will be studying regarding plastids, ribosomes, and as well as nucleus in detail. So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends. And I remember you people to subscribe my channel for getting more and more videos what I make day to day. Thank you.